Discovery number 6 is that molecular machines and sophisticated software algorithms are essential to all forms of life. Two important concepts are helpful in deciding whether molecular machines and software algorithms represent informational discontinuities. The first concept is irreducible complexity. That means that multiple parts of a system are essential for a complex function, such that removal of any one of the parts would result in loss of the function. This concept was emphasized by Michael Behe in his book Darwin's Black Box. An example of this is a spark plug. As shown on this diagram, several components must be present for the spark plug to provide a spark when needed. The second concept is hierarchical coherence. This concept was emphasized by Doug Axe in his book Undeniable. This term refers to the fact that for complex engineered systems to perform a function, many parts or subsystems often must fit together coherently. For example, a spark plug has to fit into the proper place in the engine compartment of an automobile to ignite the air-fuel mixture. The spark plug must also be connected to an electrical system that sends a high-voltage pulse to the spark plug when the key is turned in the ignition switch. Many other subsystems must interact coherently for an automobile to function. Next, I'll go over several examples of molecular machines. This slide shows a cartoon of a ribosome. A ribosome is the molecular machine that translates RNA chains into proteins. To do that, it employs the tRNA molecules discussed in Discovery number 5. A link is shown on the slide to an animation describing how a ribosome functions. The ribosome is a large complex machine made up of both RNA and protein components. The ribosome must proofread the codons on tRNAs to be sure that they match with those on the mRNA strand, and must also form a peptide bond to add each new amino acid to the growing protein chain. Also critical is that the machine must translate along the mRNA chain during the process in order to process the entire message. The common core structure of ribosomes is the same in all forms of life. All ribosomes have a small subunit and a large subunit. The small subunit has the codon reading function, and the large subunit catalyzes the formation of peptide bonds. I highly recommend viewing the video, but I'll describe the basic operation here. Ribosomes have 3 distinct tRNA binding sites, A, P, and E. At the A site, a proofreading mechanism determines if the newly bound tRNA has an exact complementary match in the first 2 nucleotides with the mRNA codon. If so, then the growing peptide chain is cleaved off the tRNA in the P site and joined to the amino acid of the tRNA in the A site. While this is going on, the mRNA is clamped. After the peptide transfer reaction, the mRNA is unclamped, and a precise ratcheting mechanism moves the ribosome forward on the mRNA chain by 3 nucleotides, or 1 codon. The empty tRNA in the P site, now without its amino acid, is moved to the E site and ejected. The A site is now empty and ready to receive another tRNA. For this system to function, it requires 3 tRNA binding sites, the proofreading mechanism, the ability to catalyze the peptide transfer reaction at the other end of the tRNAs, and mechanisms for mRNA clamping and precise ratcheting. Some of these steps require energy, and that comes from a molecule called GTP. GTP molecules must bind in precise places, and another enzyme is required to react with the bound GTP to release the energy. The ratcheting mechanism involves precise conformational changes of the RNA protein complex, so that is also an essential feature of the design. The ribosome seems to be irreducibly complex because all these aspects are required. The process of assembling the ribosome is also irreducibly complex. It involves cutting of RNA strands in precise locations to make the ribosomal RNAs, and also association of the ribosomal RNAs with specific ribosomal binding proteins to create the final structure. This diagram shows the processing of RNA to form ribosomal RNAs for E. coli and for yeast. The ribosome is itself part of a much larger, irreducibly complex system of translation, including tRNAs and the enzyme systems for making the tRNAs, the genetic code, and of course the mRNAs that contain the code for functional proteins. All the parts of this system must be coherent. This slide provides some references for reviews on ribosomes. The next molecular machine that I want to describe is called ATP synthase. A cartoon of that machine is shown on this slide, and at the bottom is a reference to a video describing how it works. I strongly recommend that you watch the video before going on in this presentation. ATP synthase is a rotary motor that's coupled to an enzyme that synthesizes ATP. It's a very complex machine, and it's irreducibly complex because many parts are required for the machine to function. A proton gradient is used to make the shaft turn, and as the shaft turns, it causes conformational changes in the enzymes. Those are the green portions in the previous slide that catalyze the synthesis of ATP molecules. But you might wonder, where does the proton gradient come from? This slide shows the process for generating the proton gradient in bacteria. The blue objects are other proteins. Those are enzymes that are required to generate the proton gradient. This is a complex chain of enzymes embedded in the membrane that each depend on one another. To summarize this complex system, NADH and FADH2 generate the protons needed to run the motor, but they require the citric acid cycle. And the citric acid cycle requires another system called pyruvate oxidation. And pyruvate oxidation requires another system called glycolysis. So ATP synthase itself, a highly complex, irreducibly complex machine, must fit into a much larger system, and the entire system must be coherent. The next molecular machine that I'd like to describe is the bacterial flagellum. It's another rotary motor. This motor converts a proton gradient into mechanical motion. At the bottom of this slide is a link to a video that describes how this motor functions. This is a link to another video that describes how this motor functions. The bacterial flagellum is a very complex machine, and it's irreducibly complex because many protein parts are required to convert a proton gradient into mechanical motion. The purpose of the system is to enable the bacterium to move toward a source of molecules that the organism can use to grow on, like glucose. To accomplish this, the flagellar motion must be controlled, and the control mechanism must be connected to a sensor. and the sensor must distinguish between molecules. This slide shows an image of that sensor system. This slide shows an expanded view of the sensor system and also how the sensor communicates with the flagellar motor. The point, again, is that the flagellar motor is one part of a much larger system, and all the parts of the system have to be coherent. The final machine that I'd like to describe is a motor protein system called kinesin. This slide provides a link to a video that describes how that machine functions. Kinesin is a machine that moves cargo along microtubules within cells. To perform that function, it has to do several things. It must convert chemical energy to mechanical motion. It must bind to microtubules in the correct orientation. Of course, it has to bind to the cargo, and it also has to release the cargo and also release itself from the microtubule. Kinesin is irreducibly complex because many complex parts are essential to carry out its function. But kinesin motor proteins have to fit into a much larger system involving microtubules and cargo components. And also the system must have a need for this directed transport. The entire system must be coherent. Regarding other machines that we're more familiar with, we know that physics, chemistry, and engineering principles can explain how they function. But the same principles tell you nothing about where the machines came from, because for each case, the source of the machine is a mind. They are informational discontinuities with respect to unintelligent natural processes. Shouldn't the same hold true for molecular machines? Molecular machines are essential to all living systems and seem to be enormous informational discontinuities. 100 years ago, no one could have imagined that molecular machines would be found at the core of all living things. Next, I'll describe another recent remarkable discovery of science, that all cells have sophisticated software algorithms. This slide shows an example of a simple algorithm carried out by molecules. It concerns the lac operon. This was the first well-understood example of gene regulation. You're probably wondering how algorithms can be carried out by molecules. The way this happens is that the binding of one molecule, say a protein, to another molecule, either another protein or DNA, either allows for or blocks the action, usually binding, of other molecules. So the algorithm is built in by the highly specific and conditional nature of the molecular interactions. I'll go over this example in some detail so you get the sense of this. The lac operon is the genetic regulatory system for controlling the production of a series of enzymes that enable E. coli to break down lactose. Lactose is a sugar in dairy products like milk. But E. coli prefers to use glucose because it requires fewer steps and less energy to break down than for lactose. The system is regulated so that the series of lactose enzymes are only produced when lactose is present and when glucose is not present, so as not to waste energy and other resources unnecessarily. The blue lines indicate the relevant section of DNA. The smaller green circle represents a protein called the lac repressor. When it binds to the DNA, it blocks the action of the machine that transcribes the genes into RNA, called RNA polymerase. This is negative regulation. The red circle is a protein called CAP. When it binds to another location on the DNA strand, it enables RNA polymerase to bind and transcribe the genes. This is positive regulation. RNA polymerase doesn't transcribe well without the help of CAP. Again, the lactose enzymes are turned on only when two conditions are met, when lactose is present and when glucose is not present. So the system includes sensors for both glucose and lactose. When lactose is not present, the repressor protein is produced and it binds and blocks RNA polymerase. But when lactose is present, it's converted into another molecule that binds to the repressor and causes it to release from the DNA strand. So the repressor itself is regulated by the presence of lactose. The lac operon is also regulated by the presence of glucose. When glucose levels are low, another molecule is produced called cyclic AMP or CAMP. CAMP binds to CAP and enables it to bind to the appropriate site on the DNA strand, thus promoting transcription. When glucose levels are high, no CAMP is made, so CAP can't bind and promote transcription. For further information, please see sites discussing the lac operon on the internet. That was a very simple example. In general, gene regulatory networks are much more complicated. This slide from a U.S. Department of Energy website shows a generic regulatory network. A signal from outside the cell is detected by receptors on the surface of the cell. That stimulates the action of a cascade of enzymes, such as kinases, that activate other proteins called transcription factors. The transcription factors bind to DNA in ways that promote transcription of certain genes. The genes that are transcribed can lead to proteins that enable cell functions, but can also generate proteins that provide feedback to further regulate gene transcription. The situation is actually much more complicated because fairly recently it has been discovered that various forms of RNA play important roles in gene regulation in addition to proteins. This slide shows an image from a common cell biology textbook that indicates that typically a large number of proteins are involved in the regulation of a gene. Further, the DNA sequences that regulate transcription of a gene can be close by or very far away from the gene. This is an image from a review article on the genetic regulatory network controlling the cell cycle in a genus of bacteria called Colobacter. A tremendous amount of very impressive work has gone into unraveling this complex network. It looks like the diagram of an electric circuit in a microprocessor. Keep in mind the fact that a basic characteristic of any living system is that it can reproduce, so some form of a cell cycle is essential for life. The paper provides the following details. There are 550 regulated cell cycle genes. 586 RNAs are also critically involved. The system is hardwired to turn genes on and off. Chromosome replication acts as a clock, controlling the timing of certain events. DNA methylation plays a critical role. DNA methylation is an example of a modification of DNA that changes gene regulation without changing the DNA sequence. The authors describe this genetic regulatory network as a robust system with many regulation modes. I encourage you to take a look at the paper for a deeper appreciation of this amazing genetic regulatory network. Genetic regulatory networks in multicellular organisms are even more complicated. This diagram is from the website of Eric Davidson and co-workers, who worked for many, many years to unravel the developmental regulatory network in sea urchin embryos. I'll cycle through several slides to give you a flavor of how the system functions. Each slide is for a different time during the developmental process. Each color represents a different layer in the embryo. The different layers develop into different tissue types. Remember, all the circuit elements turn on and off because of precise and conditional interactions between proteins, RNAs, and regions of DNA. Based on discoveries 5 and 6, some highly knowledgeable people have changed their views about design. Antony Flew was an atheist who debated theists for much of his professional career. But near the end of his life, he announced that he had changed his view and that he believed that design was real. Michael Behe is a professor of biochemistry at Lehigh University. When he began his career, he accepted the idea that Darwinian processes of mutation and natural selection could explain the working of the cell. But as he began to think about this and investigate it in more detail, he came to the conclusion that the theory is far too inadequate and that design is real. Gunter Beckley, who unfortunately passed away recently in an accident, was an accomplished paleontologist who also began his career believing that mutation and natural selection could explain life. But after investigating the question more closely, he became convinced that life was designed at some level. I encourage you to look into the stories of these three men. Others see the same evidence, but don't conclude design. But the difference doesn't come from a disagreement about the evidence itself. Thomas Nagel is a very high-profile American philosopher of the mind. In 2012, he published a book titled Mind and Cosmos. I highly recommend that book. While rejecting the adequacy of neo-Darwinism to account for the molecular basis of life, he also stated his resistance to design. He said this, "I confess to an ungrounded assumption of my own, in not finding it possible to regard the design alternative as a real option." Richard Lewontin was a highly regarded American evolutionary biologist. He certainly had extensive knowledge of the molecular basis of life. At one point, he wrote this about his commitment to materialism. He said that commitment is absolute, for we cannot allow a divine foot in the door. Dennis Noble is a British physiologist and biologist. He played a leading role in developing models of the heart, from genes to cells to the entire organ. He's a leading figure in the movement called the Third Way, which will be discussed in Discovery number 7 on our list. He's written several excellent books that I highly recommend on the inadequacy of neo-Darwinism, and advocating for the systems approach to biology. In one of these books, he mentions a discussion with his colleague about his concerns regarding the inadequacy of the neo-Darwinian conception of life, and he says that the colleague said this: "I would go along with you, Dennis, except that what you're arguing would let God back in." Paul Davies, in his book The Goldilocks Enigma, which I highly recommend, referred to the same concern, speaking of the dreaded T word, teleology. He goes on to say teleology, or final causation, is taboo in orthodox science. So you see that there's no issue with the data of science. There's no disagreement about these discoveries, but rather there are differences of opinion about the philosophy of materialism, about whether causes outside of nature can be considered. But this is a personal philosophical decision. It's not about science versus religion. Well, how bad can it get for a materialist before a cause outside of nature could be considered? One author writing in the journal Nature said this: "Even if all the data point to an intelligent designer, such a hypothesis is excluded from science because it's not naturalistic." So for some people, no amount of evidence would ever be enough. But I feel strongly that this is a personal decision, and people holding that perspective should not impose that opinion on others. To summarize this discovery, it is now known that molecular machines and sophisticated software algorithms are essential to all life forms. Many systems are irreducibly complex. Molecular machines and sophisticated software algorithms are integrated such that each entire system is coherent, characteristic of engineered systems. These discoveries seem to represent enormous informational discontinuities with respect to unintelligent natural processes. No one knows how these systems could have come about. You might wonder, is there any evidence that suggests that all of this could have come about by random mutation and natural selection? There seems to be no way to directly test that, because as of now, there's no such thing as a replicating, self-sustaining chemical system to set up to see if it can produce molecular machines or algorithms. However, it is possible to test and understand the capabilities and limitations of natural selection in living systems. The next important discovery on our list, number 7, is that such studies have shown that natural selection has severe limitations as a creative mechanism. So the combination of the discoveries of the profound informational discontinuities at the core of life, the information processing system, the molecular machines, sophisticated software algorithms, along with the discovery of the severe limitations of natural selection as a creative mechanism, has led to a profound crisis in evolutionary biology that'll be discussed in the next discovery on our list.